Welcome to this explainer. Today, we're diving straight into the fascinating link between molecular biology and complex eye diseases, specifically inherited retinal dystrophies. We're going to look closely at how the absolute smallest changes in our DNA can have massive impacts on our vision, and more importantly, how understanding those tiny, microscopic changes is literally unlocking incredible new treatments. So, what if a single typo in your DNA could steal your sight? In the realm of inherited retinal dystrophies, this isn't science fiction, it's a daily reality. But I don't want you to think of these as just static medical diagnoses. Instead, think of this explainer as a medical detective story. We're going to track down the microscopic culprits that cause blindness and check out the cutting edge tools scientists are using to stop them right in their tracks. Section 1: The Retinal Genetics Mystery: Phenotype versus Genotype. Let's get right into it. The fundamental issue for doctors here is that diagnosing a retinal disease clinically, well, it just doesn't tell the whole story. We have the phenotype, which is what we see, the clinical diagnosis and the physical symptoms a patient actually experiences. And then we have the genotype, the molecular cause, the actual underlying code in the DNA. So why does this matter for us? Well, because in inherited retinal diseases, you could literally have two people sitting in a waiting room with the exact same physical symptoms, the exact same phenotype, but they might have entirely different genetic causes. A single clinical label definitely does not mean a single genetic reality. This complexity is perfectly illustrated by a concept called allelic heterogeneity. That's basically when different mutations living inside the exact same gene produce entirely different clinical manifestations. So, two patients with a mutation in the exact same gene might experience drastically different realities. We're talking different ages of onset, varying disease severity, and completely different rates of vision loss progression. It's like a giant puzzle. Moving on to Section 2, Suspect 1, Retinitis pigmentosa. Retinitis pigmentosa, or RP for short, primarily targets the rod photoreceptors. This means the early warning signs are usually poor night vision, followed by a slowly constricting visual field. When doctors examine an RP patient, they're typically looking for this classic clinical triad: peripheral bone spicule pigmentation, a narrowing or attenuation of the retinal arterioles, and a paleness to the optic disc. But wait, remember our genotype versus phenotype rule from earlier? The inheritance pattern for RP varies wildly. It can be passed down as autosomal dominant, autosomal recessive, or X-linked recessive. It can even be part of a much broader systemic issue like Usher syndrome, where it's actually linked with deafness. So you've got this one clinical triad, but dozens of potential genetic paths. You can think of the visual cycle like a precisely calibrated engine. First, light hits a protein called rhodopsin. Second, this triggers a phototransduction cascade of signals. And third, the retinal pigment epithelium, basically the support cells, have to recycle the visual pigment so the engine can keep running. Now, back in 1989, scientists actually found a mutation in the rhodopsin gene that literally breaks this machine. Today, we know of over 100 different mutations in this visual cycle that all lead to the exact same phenotypic endpoint: retinitis pigmentosa. It's a broken engine, sure, but there are over 100 different parts that could be the culprit. All right, Section 3, Suspect 2, Stargardt disease. If RP is the thief of peripheral vision, Stargardt's disease is a juvenile macular dystrophy that severely impacts central vision. And guys, the genetic scale here is staggering. Over 900 known disease-causing mutations have been discovered in just one single gene, the ABCC4 gene. That one gene is responsible for the vast majority of Stargardt's disease cases. I mean, just imagine trying to find the one specific error among 900 possibilities in a patient's DNA. The impact of those mutations is severe. We're looking at characteristic yellow-white pisiform flecks in the eye, progressive macular atrophy, a really devastating loss of central vision, and intense photophobia. But what's actually happening on a molecular level? Well, the defective ABCA4 protein basically fails to do its job in the visual cycle. This causes a toxic, lipofuscin-rich material to build up. Over time, this toxic accumulation literally poisons and damages the retinal cells. It's a rough process. Section 4, the Detective's Toolkit, Genetic Testing. Let's see how we solve this. Using DNA extracted from something as simple as a blood draw, a buccal swab, mouthwash, or even just a single strand of hair, geneticists bring out the heavy machinery. We're talking about whole exome sequencing, or WES. This technique analyzes the protein-coding portions of the entire genome. Because so many diseases originate right in these coding regions, WES allows doctors to scan vast amounts of DNA all at once. Alternatively, they might use gene panels. These are great because they efficiently sequence a specific, targeted list of candidate genes known to cause a particular phenotype, doing it all simultaneously. So why is mapping the exact typo so incredibly important? Well, for sure, it confirms the diagnosis and allows for accurate genetic counseling so families can understand the recurrence risks. But the real game changer is this. Knowing your exact molecular mutation is now the literal ticket to getting into genotype-specific clinical trials. Your specific typo basically dictates your specific cure. How amazing is that? Which brings us to Section 5, the resolution emerging retinal therapies. Curing genetic blindness used to sound like pure science fiction, but gene replacement therapy is making it a clinical reality today. Here's how it works. Step 1. Scientists package functional, healthy DNA into a vector, usually a modified virus, because, let's face it, viruses are nature's absolute experts at breaking into cells. Step 2. This vector is delivered directly into the targeted retinal cells. And Step 3. The cell starts reading the new DNA and expressing the missing or previously defective protein. And this isn't just theory. Landmark success was actually achieved using this method to treat RPE65 mutations in dogs. It completely restored their ability to navigate obstacles, which successfully paved the way for human trials. Similar trials using lentiviral vectors are also underway right now for that massive ABCA4 gene we were talking about earlier. But what happens if the disease has already progressed and the target photoreceptor cells are just lost? Well, science is turning to stem cell-based treatments. Instead of trying to fix broken code, this strategy surgically implants entirely new retinal pigment epithelial precursor cells right into the submacular space. This aims to replace or support the tissue that's already been damaged. This brings us to a really defining truth. Diagnosis and treatment are becoming increasingly linked. Historically, identifying a gene was purely academic. It just gave you a name for the disease. But now it determines if a therapy is actually going to work. Early genetic diagnosis is absolutely crucial because these therapies rely on having viable cells left to rescue. As they say in the field, time is vision. So I'll leave you with this. If identifying a single altered DNA sequence can unlock the cure for blindness, how will genetics redefine the rest of medicine? The eye is definitely leading the way right now, but this highly targeted genotype-specific approach is for sure the future of all healthcare. Thank you so much for joining me for this explainer. Keep exploring and never stop asking questions.